Hi everyone, welcome to Unit 3, Chapter 3, where we will be learning about gene expression. Now, we already learned how genes are inherited from both parents. Now we're going to learn about how gene is expressed, mainly through the flow of genetic information as we go from DNA to protein synthesis. As always, if you click on the book, you will get a copy of the guided notes, which you can later use for extra credit. Now, before we begin, let's talk about the history of why we know what we know. Namely, the very first accurate model of DNA, or deoxyribonucleic acid, was proposed in 1953 by James Watson and Francis Crick. You may have heard of the Watson-Crick model. Now, this discovery was only possible through the process of X-ray crystallography, or X-ray diffraction. X-ray diffraction is when we take a strand of DNA and we shine an X-ray through it. Across the room, there is an X-ray reactive film, and this allows the image to be enlarged many, many times. So Watson and Crick took this image and they noticed this distinctive X shape. But what they proposed or hypothesized was that this isn't an X shape, it's a strand, and it's actually two strands that are twisted together, and the X is just where they combine. They also noticed these dark areas, and they proposed that these are the nucleic acid base pairs, adenine, cytosine, thymine, and guanine, and those base pairs are connected by something they called Watson-Crick bonds. We now know that those are hydrogen bonds. The pair actually won the Nobel Prize in medicine along with someone named Maurice Wilkins. They all worked together at Cambridge University. They also worked with someone named Rosalind Franklin, who worked very closely with Wilkins. Now, there is speculation that Wilkins and Franklin did not get along very well, but Wilkins is actually the one who pioneered the X-ray crystallography and showed Rosalind how to do it, because Rosalind Franklin was actually a chemist. But Rosalind Franklin was the first one to take a photograph of DNA called Photo 51. Here it can be seen with that distinctive X shape. This was Rosalind's discovery, but she never published her findings. Actually, Wilkins went into her notes and showed Photo 51 to Watson and Crick without Rosalind's knowledge or permission. Now, most of this history is been speculated, but Rosalind was not awarded the Nobel Prize along with her colleagues, mainly due to the stipulation that it can only be shared among 3 people. And Watson, Crick, and Wilkins already took credit for it, since Rosalind actually died of cancer before she could protest. Later on, a rule was implemented that the Nobel Prize can't be awarded posthumously, but that was not the case at the time. Now here we can see the Watson-Crick DNA model. We see that distinctive helix shape, and you can actually see that there's like a large curve and then a small curve. This isn't important for us, but it actually does help enzymes bind to the DNA at that larger curve. Now we notice that we have 4 nucleic base pairs or nucleotide base pairs. We have cytosine, which is always bound to guanine, and thymine, which is always bound to adenine. Notice that some of them have 2 rings. These are called purines. Adenine and guanine are both purines. They're much larger than their partners, pyrimidines. This will prevent incorrect base pairing because just the size. We could not have guanine bind with adenine because it would make it too wide, creating an unstable DNA connection. We can't have cytosine bind with thymine because they're too small and the hydrogen bonds would not connect. Now, we also might notice that there are some dotted lines here, and these represent those Watson-Crick bonds, or as we call them, hydrogen bonds. Notice that cytosine and guanine have 3 locations for hydrogen bonding, whereas thymine and adenine only have 2 locations for hydrogen bonding. That prevents any mismatching between thymine and guanine and cytosine and adenine. So we have this kind of model. The other parts of the DNA are going to be the ribose sugar and the phosphate backbone. So we alternate phosphate ribose, connecting the 2 strands as a polymer. We also notice that we are always going to go from the 5' to the 3' direction. Well, what does that mean? It means that these sugars are all pointed this way, and the other strand is anti-oriented with these sugars all pointing this way. Well, what do I mean by the sugar pointing? Well, each sugar has 5 carbons. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And any polymer or enzyme is always going to move from the 5' to the 3' direction, this way. So we always say that the sugars are pointed from the 5' to the 3' direction. Looking at these nucleotides, looking at these nucleic acids, which one represents guanine? Well, the first thing that I notice is that this has 2 rings and this only has 1 ring. This has 1 ring and this only has 2 rings. So I can automatically eliminate the ones with only 1 ring because I know guanine is a purine. The other thing I notice is the hydrogen bonds. There are 2 hydrogen bonds here and 3 hydrogen bonds here. I know that guanine and cytosine connect with 3 hydrogen bonds, so that means that this one must be guanine. Which one is thymine? Well, just the opposite. We are looking for pyrimidine, so it's either going to be this one or this one. Notice that we have 3 hydrogen bonds, which we know are only between guanine and cytosine, and 2 hydrogen bonds always between adenine and thymine. So I know that this is thymine. If we were talking about RNA, I would label it uracil or with a U, because thymine and uracil are shaped very, very similarly. Now let's talk about actual gene expression. So the proposal is that one gene or one strand of DNA from start at the 5' to stop at the 3' is going to code for one single trait or a specified enzyme. So in the 1920s, George Beadle and Edward Tatum decided that they're going to call this the one gene, one enzyme hypothesis. Now that isn't exactly true because it's not always an enzyme. So we now call it the one gene, one polypeptide, and we still credit it to George Beadle and Edward Tatum. But how do we create a polypeptide? Well, it happens in 2 main steps. First, we go through transcription. Now, when I'm transcribing something, I transcribe my notes. For instance, I'm going to a lecture and the lecture is being taught in English. So I'm going to take my notes in English, but it's not going to be word for word exactly what the instructor says. It's going to be shorthand. So mostly the same, but maybe a few small changes. That's just like going from the nucleic acid language of DNA to the nucleic acid shorthand of RNA. I haven't changed the language from a nucleic acid language. I'm still writing in the same language, but think of RNA as the shorthand. And this always happens inside of the nucleus because remember, DNA cannot leave the nucleus. So inside the nucleus is transcription. The second step is called translation. Let's say that my friend only speaks Spanish and they wanted to know what the lecture was about too. So in this case, I'm going to translate my notes, my shorthand notes, into the language that they understand, which in this case, my example is Spanish. Translation is going to be happening outside of the nucleus, either in the cytoplasm or more likely in the endoplasmic reticulum, pretty much anywhere that we find ribosomes. Now, translation, I'm going to be changing languages. So I'm going from a nucleic acid language to a polypeptide, which is an amino acid language. Silly little A's there. So 2 main steps, transcription, where I go from DNA to RNA, and translation, where I go from RNA to polypeptide, which after development will be a protein. And that protein could be anything. It could be structural, it could be functional. It doesn't matter what kind of protein it is, but one gene from start to finish, start to stop, will lead to one polypeptide. So let's watch this video. There's not going to be any sound. If you would like to watch the video with sound, you can view it through the PowerPoint, but let's just observe and see what we're talking about. So here they're showing us inside the nucleus is a strand of DNA, and we measure one gene as an area from a promoter or the start location to the terminator or the stop location. So we see the promoter and the terminator region. And this is considered one gene. This will lead to one polypeptide. So here we have a protein. It's all folded up. How do we get there? Well, we're going to get there in two main steps. The first step is going to be transcription. It happens inside the nucleus. The second step is translation, which happens outside of the nucleus. During transcription, we're not changing the language. We're still using nucleic acids. During translation, we take the nucleic acids and we use them to translate into amino acids. See, here we have the amino acids binding together. All right, let's see if we remember which one is it, transcription or translation. It says, what is it called when a gene is copied from DNA into RNA? Have we changed the language? No, we're still using nucleic acids, so we're going to call it transcription. And transcription happens in the nucleus. What does it called when we go from RNA to amino acid polypeptide? Well, we're changing the language. We're going from nucleic acid to amino acid. So this is translation, and it's going to happen outside the nucleus, usually in the endoplasmic reticulum. Let's talk about transcription. Now, transcription only happens in the nucleus because DNA cannot leave the nucleus. There are some main players, mainly RNA polymerase and this polymerase complex. So it's kind of multiple enzymes working together to transcribe the DNA. This is always going to happen in the 5' to 3' direction. So we're focusing on this top strand here, this template strand. Okay, so we're going in this direction, and we're always going to start at the promoter. Now, the promoter doesn't actually code for anything, but it contains a repeating pattern of TA. We call it the TATA box. This just is a really easy place for the polymerase to recognize and bind to. The first thing to bind is transcription factor, which will later assemble to this large polymerase. But the transcription factor recognizes that TATA box and opens up a replication bubble. Now remember, when we talked about DNA replication, we called it a fork. But this is not a fork. It doesn't completely open. It's just a bubble. And we're always going to go from the 5' to the 3' direction. Once the bubble opens, then we go to the process of elongation. So this whole image right here is just talking about initiation. How do we start the process? So it happens in 3 steps. The first step is initiation, and we already talked a little bit about initiation. RNA polymerase complex is going to assemble at the promoter and cause a little transcription bubble to open up. Okay, we're going to transcribe in the 5' to 3' direction. It's going to be a direct copy of the coding strand here, but we'll actually be moving along the template strand. So the second step is elongation, where RNA polymerase is going to transcribe in the 5' to 3' direction inside the DNA bubble. It's called elongation because the strand of RNA just kind of like slips and it gets longer and longer as polymerase moves in this direction. And of course, it's transcribed in the 5' to 3' direction. Now, we're going to continue in the 5', 3' direction until we reach some sort of stop point or terminator. When the transcription bubble reaches the terminator, it causes the polymerase complex to disassociate or disassemble. All these little enzymes that came together to create this complex will now come apart. And that's a good thing because then they get to reassemble at a new TATA box and go be recycled. And what we're left with is a perfectly transcribed RNA strand. So during transcription, we're going to take the DNA and we have to recognize the TATA box or the promoter region. This is where RNA polymerase complex will assemble, which is made out of several different enzymes. So during initiation, we see the promoter region and the enzyme will attach there. Then it will open up the DNA into a little bubble. Let's see the bubble. Once the bubble is open, it starts matching base pairs. Notice that we're not changing languages. We're still talking about nucleic acids. It's going to elongate, or this string is going to get longer from the 5' to the 3' direction. This will eventually become mature messenger RNA or mRNA. It's not quite mature yet though. Right now it's just a copy, a transcription, until we reach the terminator region, and the terminator region kind of stops the enzymes and causes the enzymes to fall apart, which like I said is great because they can then be recycled. Now, let's see if we can remember what we're talking about. This says the polymerase complex hits a stop point and releases. It disassembles to be recycled to transcribe some other new gene. Is this talking about the initiation, elongation, or termination? Well, it's a stop point, so it must be step 3, termination. This one says the RNA complex assembles at the promoter, which is usually called the TATA box. So is this going to be initiation, elongation, or termination? Well, since it assembles at a promoter, I'm going to call it step 1, initiation. This one says RNA transcription occurs in the 5' to 3' direction and inside of a DNA bubble, and it moves forward. The new RNA strand gets longer, so this must be step 2, elongation. Now, I already mentioned that this isn't quite mature RNA yet. We still call it mRNA or messenger RNA, but it's unprocessed. It still cannot leave the nucleus. For it to be able to leave the nucleus, some things have to happen. So DNA is made up of different sections called exons and introns. Only exons actually code for any genetic information. So what we want to do is we want to cut out the introns and remove them. This is called RNA splicing, and it happens naturally with spliceosomes, little enzymes that like to connect to the intron areas. You might wonder why eukaryotic cells have introns, because prokaryotic cells do not. Prokaryotic cells skip this stage, but they also don't have a nucleus, so there's nothing preventing them from leaving that region. Now, we think that prokaryotic cells have gone through DNA streamlining, meaning all the introns have kind of left over years and years and years of evolution. But really, these introns are important for our DNA. We need the introns. There are various theories. One of the theories is that these intron regions then become other types of RNA, like tRNA or snRNA, other types of useful RNA. Another theory is that when we have these non-coding regions and there is damage to the DNA, it is less likely that errors happen in the coding sections of the DNA because we have so much non-coding DNA kind of protecting us from damage. So those are two really good reasons why we have introns, but we don't need them for DNA processing. We don't need them for protein synthesis. So we take them out in a process called splicing. The next thing we need to do is give the RNA permission to leave the nucleus, and the only way is by adding a 5' cap. And this is a guanosine cap. It's a methylated guanosine cap. It sounds a lot like the base pair guanine. That's because it's shaped exactly like the base pair guanine, except that it's methylated. So it's very similar, and I'll show you a picture in a second. The next thing that has to happen to give the RNA permission to leave the nucleus is add a poly-A tail. This is on the 3' end, and it's literally just a whole bunch of base pairs adenine kind of repeated a whole bunch of times. And this seems to help protect the mRNA from damage, because if damage occurs, we might lose a couple adenosines, but that doesn't really matter because we have so many of them. So these are 3 different things that has to happen to the RNA before it's allowed to leave the nucleus. So here, I told you I would show you a picture of the guanine cap. This is methyl guanosine. It looks just like regular methyl guanosine, or just looks like regular guanosine, but it's kind of anti-oriented. And instead of being connected to 1 phosphate like we normally are, it's connected to 3 phosphates. And it's a 5' to 5' bridge, meaning instead of being 5' to 3', it's kind of flipped backwards, 5' to 5'. Now, the other thing is taking out these introns happens with an enzyme called spliceosome, which is actually a complex made out of 4 mini spliceosomes. They just are attracted to that region, and they connect to that region and then fold up and detach, perfectly attaching the 2 exons together. Okay, and this creates that mature mRNA. Now we're ready to leave the nucleus. So this video doesn't actually show the 5' guanosine cap or the 3' poly-A tail, but it does show us the exons and introns and how the spliceosome connects to the RNA intron and then curls up and separates. So, here we have the 5' cap and the poly-A tail. They're not super detailed about that, though. They are detailed about these spliceosomes. They just assemble on this region, curl up, and then detach. So now we have what's called mature mRNA, and that poly-A tail and guanosine cap allow it to leave the nucleus. Now we can undergo translation inside the endoplasmic reticulum. This says list 1 way that transcribed RNA has to be modified in order to leave the nucleus. Well, we could splice out the introns, or we could add a poly-A tail and a 5' guanosine cap. So that's always on the 5' end, and that's always on the 3' end. Notice that the mature mRNA is much shorter than the region of DNA that we copied. All right, next we're going to talk about translation. Translation is achieved by the ribosome. Now, we already talked about ribosome when we talked about parts of the cell. It is a non-membrane bound organelle that's found mainly inside of the endoplasmic reticulum, but also in the cytoplasm. And it's made up of the small subcomplex and the large complex. And sometimes it's talked about like it looks kind of like a hamburger bun with this small bottom piece and the larger top piece. But the ribosome plays a very important function in reading the mRNA transcript. So it's going to read in a triplet code, meaning that 3 base pairs will equal 1 amino acid. So for instance, AUG is a codon, and it will be matched with the anticodon UAC. Each of these is paired with a unique amino acid. So there are actually 64 different copies, 64 different codons that could code for 64 different anticodons. but it actually only carries 20 different amino acids. So there's only 20 amino acids that it could carry for. So there's a lot of redundancy, which actually helps in case there's a mutation. You notice that the first base pair, we have U here, and then we will go to the top and find the second base pair, which is U, and then we will go to the side and find the third base pair, which is U. If this third base pair has a mutation, we probably will still get the same answer, unless the mutation is really bad, and then we'll get a different answer. But this redundancy of having 64 codons for only 20 amino acids means that there's 4 different codes that could possibly code for leucine. There's 4 different codes that could possibly code for serine or proline. And this really helps with redundancy in case there's an error. There are also 3 codons that do not code for any amino acid at all. They are stop codons, and they actually code for termination. Then there is only one start codon, and it does code for methionine. So we do need to pay attention to that as well. So what are these thingies that are carrying the amino acids? Well, these are called tRNA, and they will have an anticodon oriented towards the bottom, and they will always connect to a specific amino acid on the top. These tRNAs, or transfer RNAs, do a very, very important job, and they are recognized by the ribosome. They are a short clip of RNA, so they are made out of RNA, but they are shaped in this weird shape because of these hairpin loops. So it goes back and connects to itself quite a bit, and then it means that this bottom region is saved for the anticodon, and the top region is the amino acid attachment site. So there are 64 different tRNAs, and only one tRNA will match one codon. We do need to know how to read charts like this. So the first base pair is going to be listed on this side. So if I have a codon that is UUU, I will follow the first, then the second base, then the third base. UUU, and I will get this one. Like I mentioned, there are 20 different amino acids, and there's nothing really more to say about this than what we talked about in the first unit when we talked about amino acids. Each amino acid is made up of an amine group, which is a nitrogen, and a carboxyl acid group, which is a carbon with a hydroxyl on it, okay? And then a central carbon, which is connected to an R side chain. Now, there are 20 different side chains, which will cause the amino acid to have different characteristics, either non-polar or polar, or a positive charge or a negative charge. For instance, tryptophan has this shaped R side chain and is non-polar. Now, let's talk about the ribosomes. We have the small subunit and the large subunit. Those subunits are separate, and they will assemble around the start codon AUG. Okay, so they will assemble around AUG. And then the specific tRNA that is carrying methionine, there's only one. If we go back and look at this chart, there's only one that codes for methionine, which is AUG. Once that tRNA is in place, we can begin translating in the 5' to 3' direction. We're always going to go in the same direction. Now, there are 3 main sites, binding sites on a ribosome: the A site, the P site, and the E site. It's APE, so it's easy to remember. The A site, I say, represents amino acid, because the amino acid will come into this place and kind of try itself out. If the tRNA matches the triplet code, then it'll stay. If the tRNA does not match the triplet code, it will be rejected. Once it finds a match, the whole complex will kind of move over, and that triplet or that tRNA will now be in the P site. The P site stands for peptide bond. This is when the amino acids will be connected to the next amino acid with a very strong peptide bond. And what we'll actually see is this long chain amino acids starting to form out of the P site. Now, once this happens, it will disconnect from this tRNA and it will move into the E site. The E site stands for exit. And that's when the tRNA, which is now naked and no longer has an amino acid connected to it, will then be rejected and recycled. It will go find another amino acid to connect to. But this long polypeptide chain will continue to grow out of the ribosome until we reach a termination point. Here you can see this happening on a single RNA strand. We have the 5' to 3' end, which means all of these ribosomes will be moving in the same direction. So multiple ribosomes can be translating at the same exact time. We see this one is at the beginning of its translation, and then this one is close to the end of its translation, and it has this nice folded polypeptide. And they can do this continuously. Sometimes it's called beads on a chain because it looks kind of like beads on a chain. Here's a more dynamic image than the last one where we can see the A site is kind of trying out different tRNAs until the anticodon matches the codon. The P site, we can see the polypeptide bonds forming between all of these amino acids, leading to this long polypeptide chain. And then we can see the E site where the empty tRNA is now rejected and it will go find a new amino acid to connect to so that then it can be recycled. This is a really great video. If you would like to watch it with sound, you can watch it through the PowerPoint, but we can actually see translation happening in this model. So here we have our codon. And it's showing you the codon chart. Again, we do need to know how to read the codon chart. We will always be looking for methionine, the start codon. So here we have AUG, the start codon. It codes for methionine. And at the end, there's going to be a stop codon. We'll get to that part. So the ribosome will assemble around the start codon, AUG. And this is because the tRNA that is specific to the amino acid methionine will bind to that location. They're showing you the anticodon that matches the codon and the tRNA holding methionine. This will cause the upper ribosome large subunit to connect. And now we are starting at the P site. The A site is empty, so other tRNAs will come in and try to match their anticodon to the codon that is located here in the A site. Here a tRNA is coming and it will match, leading to this amino acid being connected to the first amino acid. So now a peptide bond will form between these two amino acids as the whole complex will move from the 5' to the 3' direction. It will move over. Now this empty tRNA is in the E site and will be rejected, leaving behind the growing polypeptide chain, and a new tRNA will be added. This will continue and that chain will grow. And again, this can happen with multiple ribosomes along the same RNA. So the stop codon doesn't actually code for an amino acid, but it does code for a stop codon, which kind of stalls the whole system and causes it to fall apart, which is great because now all of those units can be recycled and reattached to a new mRNA. All right, this says when translating RNA into protein, what happens at the A site? So at the A site, new tRNAs are joined, and if the anticodon matches the codon, then the amino acid will be added to the polypeptide. Next, it's talking about the P site. Well, P stands for polypeptide. This is where the peptide bonds are formed and the polypeptide starts to grow. Next is the E site, which stands for exit. The empty tRNA will be rejected to find its own new amino acid to be recycled. And this all happens in the 5' to 3' direction. We're always going to go in the same direction. Now, so far, we've only seen the kinds of charts that are like square, and we have the first one on this side, the second one on this side, and the third one on this side. Now, I find these kinds of charts to be a little cumbersome to use. We still need to know how to use them because the test will use these square ones, but I prefer these circular ones because we start in the center and we're going to go outward. So this one says AUG. Find the amino acid which matches the codon AUG. So I'm going to start in the center, A, then I'm going to go to the second rung, U, and then I'm going to go to the third rung and find G. So here I have methionine, which I know is the start codon. That is always going to be the start codon. What is unique? Well, it's the start codon. All right, now we're looking at UAG. So we're going to start with U, we're going to start with A, and then we're going to go to G. Does that code for an amino acid? No. It codes for nothing. It codes for a stop sequence. All right, what about UGG? We're going to start with U, G, G. This is tryptophan. What about CCG? C, C, G. Now, let me ask a question. If we had a mutation at this location, and instead of CCG, it said CCA, would it make a difference? No, because CCA will still be proline. In fact, anything in this third location is redundant when I'm talking about proline, serine, alanine, valine, or threonine, or arginine. That third codon, or that third location of the codon, is redundant in many of these amino acids. And this helps prevent any changes when there's a mutation. All right, oh, here's the same question. If there was an error on G, would it make a difference? No, it would not make a difference. All right, now we have a completed polypeptide chain, but it's still not a protein yet. It's still not functional. There are post-translational modifications that need to happen. Now, don't get confused between post-translational and post-transcriptional. Post-transcription modifications look like this. This is post-transcription. So if there's a test question asking about post-transcription, I'm talking about excising introns and adding a 5' cap and a 3' poly A tail. But if I'm talking about post-translation, I'm talking about proteins. Now, the main kind of post-translational modification is folding. And we already talked about folding when we talked about proteins in Unit 1. There is a primary structure, which is just the sequence of amino acids. And due to the sequence of amino acids, we have a secondary structure, which is the basic folding due to the characteristics of those amino acids. Remember, some of them are more positive, some of them are more negative, some of them are more hydrophilic, some of them are more hydrophobic. And that will cause it to automatically kind of curl up due to these properties. Then we have a tertiary structure where it further folds up. This may happen naturally or may need assistance from a chaperone protein. Here is an example of a chaperone protein. We don't go super into detail about it, but this little top actually comes off and the amino acid goes inside this protein, kind of like a barrel, and it will protect it from different environmental changes. And that allows it to fold up in the correct pattern and sometimes even go into the a quaternary structure, which is when more than one protein combines to create the final functional product. Here I have a GIF where we can see that in the primary and the secondary structure, the protein will kind of flop around into different orientations until it becomes more stable. So the red area is less stable, and it will just find these different configurations until it reaches its most stable pattern, which will have all of those curls and pleats. The secondary organization is usually going to be beta pleating or sheeting, where it goes back and forth, or alpha helix, where it curls. Here we can see the tertiary or quaternary structure, where these small curls and folds will then join other proteins due to its characteristics or shape. It just kind of fits like a puzzle piece to create a functional protein. Sometimes it's still not functional though, and the proteins from the endoplasmic reticulum will be delivered to the Golgi apparatus in a vesicle. And as the vesicles travel through the Golgi apparatus, different things can happen, such as chemical post-translational modifications, which includes phosphorylation, acetylation, or methylation. There are many, many more changes that can happen inside the Golgi apparatus, but the main players are phosphorylation, methylation, and acetylation. So all kinds of different changes can happen to these proteins before they're actual functional enzymes. So this is showing us the completed polypeptide. It naturally folds in a given shape due to its characteristics, but it will leave the endoplasmic reticulum in a vesicle and join the lumen of the Golgi apparatus. And then these vesicles will kind of flow through the Golgi apparatus and get whatever chemical modification is in that layer. Then once it is fully modified by the Golgi apparatus, the vesicles will leave, usually going to the outside of the cell. In this case, we were talking about an enzyme that helps in digestion. All right, here we can see the beads on a chain example of the ribosomes moving in the 5' to 3' direction. We can see multiple of them translating at the same time. As it's being translated, it starts to fold up, sometimes with the help of a chaperone protein. But to be a functional protein, these modifications, folding is usually going to happen in the endoplasmic reticulum. Chemical modifications are usually going to happen in the Golgi apparatus. Here we can see the whole thing put together. Inside the nucleus, we see transcription. We are transcribing from DNA to RNA. So we haven't changed the language. We're still going nucleic acid to nucleic acid, but the RNA is shorthand. In order to leave the nucleus, we have to add a guanine cap on the 5' end and a poly A tail on the 3' end. We also have excised any introns that don't need to be there. Then that messenger RNA, the AUG, is going to bind for methionine at the P site, which initiates the ribosome complex to assemble the small subunit and the large subunit. Then we will continue to translate in the 5' to 3' direction as new tRNAs are tried out in the A site. If it matches, if the anticodon matches the codon, then a peptide bond will form between the amino acids, leading to a long growing polypeptide chain. Then the used tRNA will be in the E site, which will exit and be recycled. As always, don't forget that you have the opportunity to do the drag and drop chapter homework for extra credit. Don't forget to do the chapter laboratory write-up, where you will print and scan the QR code to watch a video of the lab. This week we're going to be extracting DNA from a strawberry. Then don't forget to take your chapter quiz. There is a practice quizzes in the lesson if you would like to practice the quiz before you take the actual quiz in Blackboard. Don't forget you also have extra study resources such as the textbook or vocabulary flashcards. Thank you so much for joining me this week, and I will see you next week.